comes in your mind when you hear the term immunohistochemistry? So let's analyze the term. Immuno, it is something associated with antibodies. Histo, that's something it is associated with the tissues or cells, and chemistry, some kind of chemical reactions. And you might be very familiar with these kind of beautiful images which are result of immunohistochemistry and are found in scientific publications and biomedical researches. But there are more to it. Antibodies and these immunohistochemical assays could be used for a wide variety of applications, ranging from cell morphology and architecture analysis, for understanding protein localization, cell division, and even cell signaling. For a wide variety of scientific usage, these antibodies could be used. Now, let's say you have a monoclonal and a polyclonal antibody. What are monoclonal and polyclonal antibodies? On a particular protein, there are many signatures, which are known as epitopes, which are bound by an antibody. If one particular antibody binds only a particular epitope and does not bind to other signatures, then it's a monoclonal antibody, right? More like a monogamous person. Now, in case of other antibodies, which can bind to several different epitopes present in the same protein, is known as polyclonal antibody. Monoclonal antibodies are more specific than the polyclonal antibody. Gentor supplies a wide variety of monoclonal and polyclonal antibodies. Do you know how these monoclonal antibodies are generated? Let's say you want to produce an antibody against a particular protein. You inject that protein in the body of a mouse. Then you take out its B cell from the spleen, combine it with a hybridoma cell. Combine it to get a hybridoma cell. After that, you take this hybridoma cell and screen for antibodies. These hybridoma cells are antibody producing factory. Then how does the process of immunohistochemistry work? The first step is permeabilization. You used mild detergent to disrupt the cell membrane such that the antibody which you want to put inside the cell to detect your epitopes can really get in. Second step is blocking, which ensures all the non-specific binding interactions are removed and only the specific interaction is enhanced. Step 3, you add your primary antibodies. These primary antibodies detect single epitopes if it is a monoclonal antibody, or if it is a polyclonal antibody, it would be detecting multiple epitopes. Regardless, it would detect this particular protein. And next, you need to visualize the signal. In order to visualize a signal, you use a secondary antibody, which is attached with a fluorophore. In this case, this green fluorophore. Now, there are many ways by which you can detect the signal. One particular way is to use fluorescence-based method, where you use a fluorescent attached secondary antibody, and the secondary antibody is specific to the Fc region of the primary antibody. You also have colorimetric detection, where the secondary antibody is engaged with an enzyme and it can cleave a substrate to produce color. Also, there are conjugated antibodies available, which is conjugated with the primary antibody, and the detection is even easier, right? Now, the last step is to use microscopic techniques to visualize the data, and that is how your immunohistochemistry workflow works. Thanks for your attention. Stay tuned for more. Thank you.